In the summer of 1964, Tom Brock was driving through the sun-scorched basins of Wyoming on his way to his new marine lab at the edge of the Pacific, when he decided to make a little detour.
I was on my way to the West Coast, came in the south entrance.
He would remember years later.
I got out of my car, and there were these thermal areas that were just spreading out from the hot springs into the lake.
Yellowstone National Park is famous for over 10,000 naturally occurring hydrothermal features, including shallow sulfuric mud pots that smell like rotten eggs, dramatic steam vents with temperatures soaring as high as 280 degrees Fahrenheit, enormous chalky white terraces formed from rapidly depositing limestone, and about 500 geysers. However, the most common hydrothermal features are its hot springs, which form when pools of rainwater trapped beneath the bedrock are superheated by Yellowstone's underground magmatic system before rising back to the surface. Under a thermal camera, clouds of these springs flash with stunning whites and ambers and swell into rosy oranges and pinks and dark squid ink violets as the sweltering heat fans outward. But even without a special lens, Yellowstone's hot springs still look almost unbelievably brilliant, like scenes in Technicolor. Their basins teem with swarms of microorganisms that all exhibit what we call thermophile behavior. These types of creatures thrive in the scorching temperatures, and as they grow and shift and multiply, different colonies reflect and diffuse different wavelengths of light, emitting bright turmeric yellows and emerald greens and deep iridescent peacock feather blues.
I had never seen Yellowstone before. When I got there, I was stunned by all these microbes that were living in all these hot springs, and nobody seemed to know anything about them.
Every once in a while, the right person will stumble onto something important, almost entirely by accident. In 1928, the bacteriologist Alexander Fleming famously discovered the antibiotic penicillin after one of his samples was accidentally contaminated with molds from the fungal species Penicillium rubens. In 1956, the American inventor Wilson Greatbatch was an assistant professor of electrical engineering in New York, tinkering with a device he'd hoped would record heart rhythms for researchers at the university's Chronic Disease Research Institute. As he completed the circuitry, he reached into his toolbox, fumbling around for the last few supplies, and accidentally retrieved a resistor that was entirely the wrong size. When he installed it, the device emitted electrical impulses that, because of that faulty resistor, had the uncanny tempo and intensity of a human heartbeat. His little mistake would lead him to refine and debut the first implantable pacemaker. A couple years earlier, in 1945, a man named Percy Spencer, a 60-year-old self-taught physicist who worked for the massive defense contractor Raytheon, was inspecting a radar set when he noticed that a candy bar he tucked away in his pocket was melting. He was fascinated. He called for a bag of popcorn, which promptly exploded all over the lab when he held it near the radar unit. Then he jerry-rigged a kettle and set in it a single uncooked egg, shell and all. In seconds, the egg yolk shot clean away, splattering over the face of an engineer. Spencer had accidentally walked into a massive microwave. So too, in that random stop in 1964, Tom Brock accidentally discovered the species Thermus aquaticus, a bacterium that can tolerate the inhospitable conditions of a Yellowstone hot spring, and paved the way for later scientists to isolate its unusually hardy enzymes in the lab. Today, we use those enzymes to make millions of copies of DNA from a single sample at extremely high temperatures. Without this technique, we wouldn't have modern forensics. We wouldn't be able to detect and study some of the tiniest, deadliest, and most subtle genetic pathogens like tuberculosis or HIV. we couldn't efficiently quantify, clone, or sequence genetic material. If Thomas Brock hadn't wandered off course that morning, the cutting-edge tools that define molecular biology today simply wouldn't exist. And more to the point, if Thomas Brock hadn't given a second thought to those pretty colors along the water's edge, we might never have known that these types of extremophiles existed, or that countless others, including a whole new domain of life, were out there waiting to be found. I'm Michael, and this is Curiosity. In today's episode, we're exploring the secret, infinitesimal lives of prokaryotes.
Prokaryote.
Prokaryote.
Prokaryotes?
Prokaryote.
Prokaryote.
Karyote. Prokaryote.
Prokaryotes. Like so many of the words we use in science and biology in particular, this one comes to us courtesy of the ancient Greeks. The prefix pro means before, like prophylaxis or prologue, and the root word karyon means nut or seed. It's the same word from which we get kernel, and there's a theory that it might be related to the word that would eventually give us cranium or cerebrum, both associated with the brain. In biology, that word, karyon, specifically refers to the brain of the cell, the nucleus. It's the place where all our DNA is stored, where messenger RNA gets produced, and where the cell makes all its major decisions. But, as their name suggests, prokaryotes are anatomically simpler than we are. Their structures hail from a time literally before the nucleus, so they don't have one. In fact, prokaryotes strikingly lack most of the typical structures we see in the cells of other life forms, like organelles enclosed by membranes. Instead, they're basically just floating sacks of genetic material wrapped in cell membranes and cell walls and capsules. But if that's starting to sound a little familiar, slow down. As rudimentary as they seem, prokaryotes are nowhere near as basic as viruses are. Unlike viruses, prokaryotes have everything they need to replicate their DNA and process energy. They may not have membrane-bound organelles made of lipids, but they do have plenty of structures derived from proteins and nucleic acids, like ribosomes to make new polypeptides, and gas vesicles to keep aquatic cells buoyant, or even microcompartments made of icosahedral protein shells as occur in certain bacteria. Prokaryotes also have cytoskeletons similar to us eukaryotes. A cell's cytoskeleton is an intricate network of long protein filaments stretching across every square nanometer of the cytoplasm. The cytoskeleton plays a major role in determining the cell's overall shape, allowing cells to expand and contract, and even facilitating cell motility from one place to another. In eukaryotes, cytoskeletal filaments are polymers formed from actin and tubulin. Prokaryotes possess two homologs of these protein classes, called the MREB protein and the FTSZ protein. These guys help to control the cell's width and modulate constriction during cell division. Notably, prokaryotes are small, like really small. The typical prokaryotic cell has a diameter somewhere between 5 and 0.5 micrometers. Comparing a prokaryotic cell to a typical eukaryotic cell with a diameter anywhere from 10 to 100 micrometers is like comparing the size of a peewee marble to a basketball. Nevertheless, what prokaryotes lack in size, they make up for in creativity. At least bacteria do. Archaea typically exhibits simple oval shapes, or in the case of one Archaean species, a flat square. But bacteria are some of the most morphologically diverse creatures on the planet. Some bacteria form spherical cocci, like the genus of Streptococci bacteria that can cause strep throat or meningitis or bacterial pneumonia. Other bacteria, called bacilli, are long and rod-shaped. Examples include bacteria from the genus Listeria, like Listeria monocytogenes, which spreads through contaminated food and induces listeriosis in humans. Another example is the genus Salmonella, which has two famous species, Salmonella enterica and Salmonella bongori, both known for inducing the gastrointestinal disease, salmonellosis. Spirals are another major bacterial shape. Vibrio cholerae, the bacterial species that causes cholera, is one example of a bacterium that isn't typically shaped like a spiral. Instead, its shape more closely resembles one of the bacilli, or even the shape of a comma. But in a pinch, it can twist into a spiral to help it riddle its way into the linings of the gut. Other bacteria are naturally spiral. These include the spirilla and the spirochetes. A spirillum is a type of large spiral bacterium that keeps its shape fairly rigid and moves along using tufts of flagella, or tails basically, along its surface. One famous spirillum is the species Campylobacter jejuni, a bacterium that spends a lot of time squirming around in animal waste. It's responsible for the disease Campylobacteriosis. This bacterium's numerous flagella act like a secret weapon for penetrating and moving around in the intestinal epithelia of its victims. Campylobacter jejuni produces a ton of these flagella, so many, in fact, that it's had to adapt rigorous regulatory systems to keep that production in check. Just like a spirillum bacterium, a spirochete also uses flagella, but theirs are located within the layers of their membranes, sort of underneath their skin. The effect is that these bacteria tend to move in a bizarre, writhing fashion with irregular bends and twists. One common spirochete genus is Leptospira, the group responsible for the blood infection leptospirosis. Another common spirochete is the species Treponema pallidum, whose subspecies cause diseases like yaws, syphilis, or non-venereal endemic syphilis, or Begel. Spirochetes are famously slender and flexible and kind of look like corkscrews. Some of the most interesting spiral bacteria aren't strictly spirilla or spirochetes at all. These are the bacteria from the Helicobacter genus, like the species Helicobacter pylori. As their name suggests, these bacteria look a little bit like tiny helixes. They're helical. The main cell body is like a worm curling into half of a figure eight. And it achieves this unique shape through two proteins that work together to fine-tune the thickness and density of the bacterium's cell wall at key points. The helical pattern of Helicobacter pylori allows it to burrow deep into the lining of the stomach, narrowly evading the deadly reach of gastric acid in the lumen. While life-saving for the bacterium, this behavior is highly inflammatory for its victims. And it's pretty common, too. About half of the world's human population suffers from the painful effects of these types of chronic Helicobacter infections. We call them stomach ulcers. A key feature of nearly all prokaryotes, regardless of shape or size, is the presence of a cell wall. The cell wall is rigid and stable. It covers the outer cell membrane, maintains the cell's shape, protects the cell from its environment, and prevents it from bursting if it gets oversaturated with water. Aqueous solutions that are oversaturated with water relative to a cell are said to be hypotonic. In hypotonic solutions, cells without a cell wall quickly absorb water through their membranes, swell up like a balloon, and lyse open. In contrast, solutions that are low in water content relative to the cell are said to be hypertonic. In these circumstances, the environment sucks water away from the cell, causing them to warp and shrivel. When prokaryotes are placed in hypertonic solutions, their cells dry up and shrink away from the cell wall. This state doesn't always kill them at first, but it does slow down their activity, including how quickly they can reproduce. This property is why humans have slathered food in salt since prehistory, and why salt was such a valuable commodity in the age before refrigeration. Salt and similar solutes rapidly force a solution to become hypertonic, causing food-spoiling prokaryotes to lose water and inhibiting how quickly they can spread. We haven't talked about the parts of a typical eukaryotic cell yet. That's for next time. But odds are you already know that some eukaryotes, like plants and fungi, have cell walls too. These cell walls are usually made of cellulose or chitin, the two most abundant biopolymers on the face of the earth. Prokaryotes, though, are different. Most bacteria have cell walls that contain a substance called peptidoglycan. Unlike cellulose and chitin, peptidoglycan isn't made exclusively from carbohydrates. Instead, it's a rich lattice of sugar molecules and amino acids. It fully encloses the bacterial cell and grows with the cell, allowing the bacterium to retain its general shape throughout its life cycle. Archaea, always the overachievers, don't use cellulose, chitin, or peptidoglycan. Instead, they surround their cells with elaborate self-assembling protein or glycoprotein coats. The best understood of these configurations is called the S layer, or the surface layer membrane, a protective feature consisting of a single film of identical interlocking protein units. Bacteria have this particular innovation too, by the way, but in Archaea it's far more common. And cool as it is, and seriously, the S layer is pretty cool, microbiologists have historically shown more interest in the traditional component of the bacterial cell wall for two main reasons. The first reason is that peptidoglycan, the stuff that comprises most of the bacterial cell wall, is a major target of many antibiotics. And if you think about it, that makes a lot of sense. As we discussed earlier, peptidoglycan is exclusive to bacteria. Archaea don't have it, eukaryotes don't have it, and I bet if you were the first pharmacologist to realize that, it would have felt like you hit the jackpot. If you could design a drug that acts on peptidoglycan, that means you could design a drug that only recognizes bacteria, not some other innocent cell, which means you could stop a bacterial infection in its tracks without seriously harming an afflicted patient. This is the mechanism of action behind the very first antibiotic, penicillin. It works by halting the synthesis of peptidoglycan at the very last step of the process. This sort of biochemical sabotage weakens the cell wall, and as we learned earlier, a weak cell wall can't prevent water from surging into the cytoplasm. Bacteria exposed to penicillin don't just grind to a halt. They explode. The second thing about peptidoglycan that makes it so interesting to microbiologists is that it's a pretty convenient way to classify bacteria. For whatever reason, bacterial species don't all set up their cell walls in the same way. Some bacteria have a thick layer of peptidoglycan that lies right on top of their cell membrane. Others have a thin layer of peptidoglycan that gets sandwiched between the inner and outer layers of their cell membrane. We call this special region between the plasma membrane and the outer membrane the periplasmic space. If you were a medical laboratory scientist, part of your job would involve hunkering down in the basement of some hospital somewhere running tests on bacterial samples. It would be up to you to advise providers on what kind of antibiotics might work best for a particular infection. And to give good advice, it will be helpful for you to know what the cell walls of those bacteria actually look like. For instance, Say a patient came in with an infection from the second type of bacteria we just discussed, the kind with only a thin layer of peptidoglycan tucked away in the periplasmic space. Would it be helpful to prescribe an antibiotic like penicillin? Well, let's think. How does penicillin get into most bacteria anyway? It's a pretty bulky molecule. It's actually from a class of antibiotics called beta-lactams, which, if that doesn't ring a bell from organic chemistry, is a type of functional group made of a bond between nitrogen and a carbonyl that's part of a larger ring fused together with other add-ons into a 4-part structure. Penicillin is actually a special type of beta-lactam that also features another ring structure, this time with 5 parts, including a sulfur atom and some more nitrogen. So yeah, it's an oddly shaped molecule, and it's heavy, relatively speaking. Its molecular mass is over 18 times bigger than an ordinary water molecule. We've already established that water can get in and out of a bacterial cell pretty easily. Peptidoglycan keeps the cell from suddenly lysing by providing strong physical resistance to increases in pressure, but it doesn't exactly keep all that water away. Peptidoglycan is porous, it has gaps, and unlike a cell membrane, it can't pick and choose what it lets slip through. The gaps in peptidoglycan are big enough for water molecules to wriggle through, and they're also big enough for penicillin. So, since penicillin is water soluble, all it has to do is hitch a ride. By the way, this is a small part of the reason your doctor might tell you to take certain antibiotics with lots of water. Anyway, because of all of that, penicillin would be a decent option to attack that first class of bacteria we mentioned, the kind with a thick layer of peptidoglycan and only a thin membrane underneath. But if you introduced a penicillin molecule to that other class of bacteria, the first thing it would come across wouldn't be peptidoglycan. It would be an outer cell membrane. The only peptidoglycan to be found would be hidden away in a thin layer right below the surface. Not only are cell membranes generally pickier than cell walls, this particular cell membrane is reinforced with an extra film of something called lipopolysaccharide. Lipopolysaccharide is a component of the cell envelope that's unique to these bacteria. It consists of a large outer section made from a polysaccharide, that is, a carbohydrate polymer. Then it has a middle core made of a short chain of smaller carbohydrate residues. And finally, it has a special molecule with a modified sugar group, some phosphate groups, and several chains of fatty acids. This molecule is called lipid A. By the way, since we're on the subject, I should point out that lipid A is some really brutal stuff. If a bacterial cell gets ripped up by the immune system and lipid A makes its way into circulation, it can trigger severe reactions, sending a person's entire body into shock. Lipid A is one of the major causes of sepsis, for instance. It's a fierce little bacterial defense mechanism, and it's the reason why we typically don't say lipopolysaccharide outside of a basic biology course. Instead, we call it for what it really is, the endotoxin, the toxin inside the membrane of a bacterial cell. In addition to being the major bioactive component of lipopolysaccharide, lipid A is obviously a lipid, right? It has all those fatty acid chains, remember? It's a toxic little ball of fat. It's hydrophobic. Now, think back. What was one of the first things we learned in episode 1? Hydrophobic materials don't allow anything water-soluble to pass through, at least not easily. Penicillin wouldn't stand a chance. You'd be better off suggesting a fat-soluble antibiotic instead, maybe something like doxycycline. At this point, if you've been paying attention, you might be wondering how any water gets into the cell. Remember from episode 1 that all cell membranes have phospholipids, even if they don't have a special lipopolysaccharide layer above them. So in theory, all cell membranes should be pretty hydrophobic. So what gives? Hold on to that thought. We'll be digging into how submembranes bend that rule and like a dozen more in a future episode. For right now, pretend you're that medical laboratory scientist again. You know that the type of cell wall each of those bacteria has is important. You do. But how can you see it? You don't exactly have an electron microscope lying around in some storage closet. What you'll need is something indirect. Not necessarily a tool that'll let you see the cell wall, but something that will tell you where the peptidoglycan is sitting within it. In that first group of bacteria, peptidoglycan is abundant and it's right on the surface. In the second group, peptidoglycan is limited and it's trapped. So no, you can't see peptidoglycan, but who cares? What you need is a chemical that binds to peptidoglycan, something bright enough, big enough, so you can see that instead. A couple tiny but significant things happened in 1884. It was the year we saw the first edition of the Oxford English Dictionary, for instance. Actually, it wasn't really a whole edition. It was what we call a fascicle, just one installment in a larger book. Only a couple hundred pages and only had a few entries from A to Ant. And almost nobody bought it. But today, the Oxford Dictionary is one of the most definitive references of all time. It has 500,000 entries, and it chronicles over a millennium of human language. 1884 was also the year of the first real long-distance call in history.
1876, the telephone.
It was an enormously risky publicity stunt. The American Bell Telephone Company, named after the man behind the first telephone call ever, Alexander Graham Bell,
To connect every man's house
had the idea to run a reinforced telephone wire some 235 miles from New York to Boston. As one historian would later describe it, this was to be not only the longest of all telephone lines, strung on 10,000 poles, it was to be a line deluxe, built of glistening red copper instead of iron, and it would cost $70,000. That would be worth nearly $2.5 billion today, by the way, but it worked. As the story went, when the last coil of wire was stretched into place and the first hello leaped from Boston to New York, the new line was a victorious success. The Bell Telephone Group made millions and eventually evolved into a telecommunications giant called the American Telephone and Telegraph Company. Today, we just call it AT&T. 1884 was the year masons and metalworkers in Paris finished working on a special 4th of July gift for their friends across the Atlantic. That was the Statue of Liberty. 1884 was the year we decided to use the Royal Observatory in Greenwich, England, as a prime meridian to divide the globe into east and west. That's the reason we have international time zones. And finally, 1884 was the year an obscure Danish microbiologist working out of Berlin turned his attention to bacteria. He wanted to make them easier to see under a microscope, make them stand out more against the background. His name was Hans Christian Gram. Gram discovered that bacteria could be stained purple with an organic compound called crystal violet. Then that stain would disappear from certain bacteria if you washed the culture with ethanol, the chemical behind ingestible alcohol. When he exposed samples to another stain, a synthetic red dye called safranin, the colorless bacteria turned pink. Okay, what was happening? Remember what we said earlier? Peptidoglycan is porous, it has gaps. Crystal violet is great at filling in the gaps, and thanks to its intense color, it quickly turned the bacterial cell wall purple. So pretend you're that technician again and you're staring at those samples that got shuttled into the lab from a couple of patient wards upstairs. The first thing you'll need is a microscope slide, a sterile one, fresh from the packaging. Then you'll need to smear one of your swab samples across the surface and let it dry. After that, the next step is to crank up the heat. Exposing your slide to a few seconds of intense flame, like from a Bunsen burner for instance, kills the bacteria and fixes them in place. Then once that's done, you can flood them with crystal violet. In fractions of a second, the dye pools into hundreds of submicroscopic cracks, filling peptidoglycan like water in a sponge. Step 5 is to gently rinse away excess crystal violet with some distilled water. To trap the crystal violet that's already adhered to each cell, we then add a few drops of iodine. In this process, iodine functions as a mordant. It keeps the dye in place. In about a minute, that iodine will bind to molecules of crystal violet, forming big complexes within the membrane's inner and outer layers. And after that step is done, you rinse again. Then comes the first moment of truth, adding a decolorizer like ethanol or acetone or both. When we flood the slide with one of these harsh chemicals, it begins to break down the cell membrane. For bacteria with only a thin layer of peptidoglycan, the decolorizer quickly crumbles the outer membrane and leaches into the periplasmic space. What little peptidoglycan there is just washes away, and with it all the purple dye. The bacteria are stripped bare, colorless. For bacteria that have a thick layer of peptidoglycan, the decolorizer is far less effective. Crystal violet slowly escapes the cell, but not fast enough. So the cell walls of these bacteria won't lose their distinctive purple coloration. The final step is to restore color to the cells we can no longer see, the ones that weren't as resilient after being exposed to the decolorizer. This is where you'd add safranin, your counter stain. For those bacteria that only had a sliver of peptidoglycan to begin with, safranin turns their membranes pink. Those other bacteria, the ones with thicker peptidoglycan walls, the ones that were stained purple, they do absorb safranin too, but their purple color is so intense that it hardly makes a difference. Only unstained bacteria show up as pink under the microscope. And after letting the slide rest for a while, 45 seconds or so, then rinsing one last time with water, you'll be able to see the difference for yourself. Today, we call this magical little procedure gram staining. Cells with hefty outer layers of peptidoglycan turn purple and stay purple in the gram protocol. We say these are gram-positive bacteria. Other cells with only a weak layer of peptidoglycan between two fatty sheaths of cell membrane, these are said to be gram-negative. Under the microscope, they're bright pink. This innovation was small, yes, but it was significant. A full gram stain can be completed in minutes, and it's able to tell us so much information about any given sample. Importantly for you and for the provider managing patient care is the fact that gram staining is insanely good at putting medical decisions in context. Is the pathogen really a bacterium? What kind of treatment would be most effective? If the answer is an antibiotic, then which one? And would this strain likely develop resistance? Prokaryotes, like the kind that Tom Brock saw at Yellowstone, are full of life and activity and color. And they're always telling us something. We just have to know where and how to look. Bacteria come equipped with a couple more interesting features that might be worth remembering. Some bacteria, for instance, have something called a bacterial capsule. In gram-negative bacteria, these capsules connect to peptidoglycan. In gram-positive bacteria, they covalently attach to phospholipid or lipid A molecules within the plasma membrane itself. They can also interact with surface proteins on the bacterial membrane. Capsules that are more loosely arranged are also called slime layers. What are they made of? It depends on the species. In most bacteria, capsules are mostly polysaccharides or carbohydrate polymers. In others, like in the bacterium that causes anthrax, capsules primarily consist of polypeptides or protein polymers. While some bacteria, like the enormous species Bacillus megatherium, have both. And what do capsules do? A lot. They help bacteria stick to surfaces and to each other. They enhance the formation of biofilms, wide, thin, gooey matrices of polysaccharides, extracellular proteins, and small molecules. Capsules also help bacteria evade their host's immune system. They mask parts of the bacterium, or antigens, that immune cells can recognize. There's one species called Francisella tularensis that's been linked to the often lethal disease tularemia, aka rabbit fever. It's spread by ticks and deer flies, and from one infected animal to another, and it's ridiculously good at laying low for weeks in the soil or in water, or sometimes even in the carcasses of old victims before coming out to play. One of this bacterium's favorite tricks is to use its capsule to somehow impede the dispersal of an immune stimulant called R848. We're still not entirely sure how it does this, but the effects are stunning. This inhibition denies special immune cells called phagocytes the missing key they need to undergo critical metabolic transformations. Without it, they can't send out signals to other immune cells. So basically, they're stranded, ambushed by F. tularensis cells instead of the other way around. But it doesn't end there. Capsules also help bacteria penetrate one of the body's most powerful and most important defensive structures, the blood-brain barrier, or the BBB. It's a membrane unique only to the brain that very carefully monitors and restricts chemicals flowing in and out. To say that this thing is highly regulated would be a gross understatement. Almost nothing gets in. Over 90% of all small molecule drugs and nearly 100% of the larger ones simply can't cross the barrier. It's actually a major problem in the field of neuropharmacology, and one of the reasons we still don't have optimum treatment for diseases like Alzheimer's or Parkinson's. As anyone who's seen or experienced bacterial meningitis up close can tell you, certain bacteria have no problem crossing this hurdle. And for some species, like E. coli, that's all thanks to the bacterial capsule. E. coli capsules bind and invade endothelial cells that line the blood vessels in the brain. Once inside, they sneak the bacterium through the cell using some of its own specialized storage compartments called vacuoles. And it does all that while evading fleets of lysosomes, which are basically the cell's built-in trash disposal system, so that E. coli bacteria emerge on the other side unharmed. Finally, one of the most well-documented functions of the bacterial capsule is its role in enhancing resistance to antibiotics. It's the secret weapon behind some of the most terrifying bacterial species. For example, Acinetobacter baumannii, a stunningly resilient superbug that first gained attention in military treatment facilities during the Second Gulf War, has been shown to upregulate the genes that code for its bacterial capsule after it realizes that there are antibiotics somewhere in circulation. In 2023, when microbiologists modified the DNA of these bacteria to produce a mutant with a deficient capsule, they found that it became slightly more susceptible to the effects of antibiotics. Similar observations have been noted in studies of the bacterium Klebsiella pneumoniae, a species that's been known to behave as an opportunistic pathogen in humans. In other words, it preys on individuals whose immune systems have already been weakened. This puts K. pneumoniae up there with infections like MRSA and HIV, just because of its capsule. Research in this field is limited, but already researchers are raising calls for the development of vaccines to fight these bacteria, vaccines that specifically target the capsule. Whether or not pharmacologists, infectious disease specialists, and public health practitioners act on those calls may well determine what a future with bacteria will look like, and whether that future will include us too. Another protective measure bacteria sometimes employ is called an endospore. It's a dormant, tough, non-reproductive structure made of a hard outer protein coat and a thick peptidoglycan layer called the cortex. Under that is the cell wall, and under that is the cell core, which contains all the bacterium's DNA, ribosomes, and large stores of a chemical called dipicolinic acid. This dipicolinic acid makes up about 1/10 of all the weight of the endospore, and its main job is to bind with calcium ions, forming a complex that essentially sucks all the water from every layer. This reaction dehydrates the bacterium, basically mummifying it and keeping it that way for hundreds, even thousands of years. While going no contact and curling up into a ball for a few millennia might seem pretty amazing for your average introvert, this sort of behavior is pretty radical for bacteria. It's only triggered when nutrients are so low that the bacterium would risk starving to death otherwise. Endospores basically put the microbe's whole life cycle on pause, like a mini hibernation. In this state, they can survive pretty much anything: extreme temperatures, UV radiation, desiccation, chemical damage, even enzymes. And all they have to do is stay huddled tight. Then when the environment improves, so do they. And speaking of their environment, it's worth asking, how exactly do bacteria navigate? How do they anchor in place when they need to? How do they move around? The answer to the first question is something that might be a little odd to think about, but prokaryotes are hairy. Some prokaryotic cells adhere to surfaces with the aid of hundreds of short, bristly proteins projecting out of the cell envelope, called fimbriae. In pathogenic bacteria, fimbriae work alongside the bacterial capsule to allow bacteria to form colonies and to infect host cells. Under a microscope, it's pretty easy to confuse a single fimbria with a similar structure called a pilus. Pili are also protein projections, but they're longer and less numerous, almost like tentacles. To help differentiate between them and fimbriae, pili are sometimes called conjugative pili or sex pili. We'll discuss why exactly in just a moment. First, we have to talk about how prokaryotes travel. Specifically, I guess, how they swim in aqueous environments. I say that because there are a wide variety of structures prokaryotes rely on to stay motile in all sorts of settings. Some pili, called type 4 pili, can propel some bacterial species in a series of short, spasmodic jerks. We call this behavior twitching motility. There's also something called social gliding motility, which is a smooth but still pretty energy-intensive process for moving certain bacterial species over a surface using massive amounts of slime. This slime is released from the bacterium through a structure that works kind of like a nozzle. It's called the junctional pore complex. It's a huge protein complex that spans the whole width of the cell wall, and depending on which complexes it activates, the cell can strategically move itself in either direction it wishes, forward or reverse. Another set of motility structures, called ratchet structures, seem to contribute to cell locomotion in a similar way, though the details we have on these guys are so inconclusive they might as well be moving by mechanisms of chance, acting like so-called Brownian motors. That name actually comes from a physics term, Brownian motion, the asymmetric jostling of molecules. Randomness, chaos. Needless to say, the jury is still out with this idea. In at least one species that we know of, Spiroplasma melliferum, some of the smallest bacteria on Earth, motility is achieved not by some special structure on the surface of the cell, but by the cytoskeleton itself. S. mellifera has a cytoskeleton consisting entirely of 7 fibrous ribbons running the whole length of the cell. When a change occurs in the conformation of a single building block in one of those fibrils, it gets transmitted to all its neighbors and then up and up and up the chain all the way to the edges of the cell along the membrane. This forces the cell to shrink or extend, to expand or contract. And so we say this species has a contractile cytoskeleton. As interesting as those adaptations are, you probably won't run into them in your typical high school or even college biology textbook. For one thing, they're not highly conserved across that many prokaryotic taxa, just in a few species that have happened to catch scientists' notice over the years. And for another, there's so much more we don't know about how they work than how much we do. A better understanding of the diversity of their motility structures will improve how well we can plan, predict, and prepare for the spread of prokaryotes that cause disease. All information is invaluable, so we've got to start filling in those missing gaps, and fast. The motility structure that's been the most studied, and for which we have a wealth of information, is the flagellum. We started talking about flagella earlier in the episode. They're extra long, whip-like filaments that, like I just mentioned, help bacteria move through liquid media. They kind of resemble tails. Bacteria with only one flagellum at one end are said to be monotrachous. Monotrachous. Remember, mono means one, like in monogamous or monolith. Bacteria with 2 flagella, one at each end, are said to be amphitrachous. One more time, that's amphitrachous. The prefix amphi means both, like an amphibian that can go through life both in water and on land, or the chemistry term amphoteric for a substance that can work both as an acid and a base. Or, for another example, there's even a biochemistry term that we've discussed here before, amphipathic, for a molecule that has both hydrophilic and hydrophobic regions, like the phospholipids in our cell membranes. Bacteria that have a tuft of multiple flagella, but only at one end, are said to be lophotrachous. Again, that word is lophotrachous. It has a prefix that we actually don't use a whole lot in English. Well, maybe zoologists do, but not us mere mortals. It basically means a crest, or like I used a second ago, a tuft. In other words, a small arrangement. Finally, bacteria with multiple flagella spread all over the place are said to be peritrachous. Peritrachous. The prefix peri means about or around, like perimeter, the distance around a shape, or pericardium, the fluid-filled sac that sits around your heart. One more time from the top. Monotrachous, 1 flagellum at one end. Amphitrachous, 2 flagella, 1 at each end. Lophotrachous, a bunch of flagella all at one end. And peritrachous, flagella all around. The structure of an individual flagellum is pretty complicated. I won't get into it here. Trust me, you'll have your lion's share of helpful diagrams to dig into in textbooks and scientific literature and lecture halls from now until graduation day. What I will say is that it's comprised of a basal structure with a central rod, onto which is attached a hook, and from which emerges a filament. As the rod rotates, the flagellum rotates, and the rotation of the flagella determines how bacteria move through space. In peritrachous bacteria, for instance, when flagella rotate counterclockwise, they bundle together like a comet's tail and direct bacteria to run in one straight direction. But when they rotate clockwise, they're set free, and cells proceed to tumble about aimlessly until rotation changes again. It's worth noting, though, that no motion is truly aimless for a prokaryotic cell. Flagellar motility is fancy at a cost, and that cost is energy in the form of ATP. Basically, cells simply can't afford to go on a joyride just for the fun of it. Instead, about half of all prokaryotes are capable of what we call taxis, a directed movement toward or away from some kind of stimulus. This could be phototaxis, or movement toward or away from light. You can imagine how this could be useful for photosynthetic bacteria like sulfidophyllum, or most commonly, it could be chemotaxis, that is, moving along chemical currents, or even in the case of some bacteria, magnetotaxis. One of these is the species Magnetospirillum magneticum. Apparently, taxonomists thought its knack for navigating using Earth's magnetic field was so cool, it deserved a shout out in both parts of its scientific name. And, you know, they were right, it is pretty cool. I'm a cell biologist by training, so I spend a lot of time raising and nurturing and testing cell lines in the laboratory setting. You can imagine then just how tempted I am to pause here and talk about all the various prokaryotic growth requirements you need to hold your own in a microbiology lab too. We could talk about temperature specifications and the exceptions like Tom Brock's famous thermophiles. We could talk about nutritional preferences for autotrophs, the prokaryotes that use CO2 as a carbon source, or heterotrophs, the kind that rely on organic nutrients from other organisms, like glucose from plants, or chemotrophs, the kind that get all the energy they need from chemicals, or even phototrophs that get their energy from light. We could talk about growth media, and yes, God knows we could talk about growth media for ages. And cell culture vessel specifications, and how bacteria use oxygen, whether they absolutely require it like the obligate aerobes, or not like the anaerobes. And we could talk about the 3 types of anaerobes too, the facultative anaerobes that use oxygen when it's around but don't make too much of a fuss when it's not, the tolerant anaerobes that don't care either way, or the obligate anaerobes that are actually damaged by oxygen. We could talk about all of those. We could talk about the difference between fermentation and respiration and metabolism, about photosynthesis and cellular respiration. We could get messy, even get into some biochemistry. We could talk about bacterial life cycles. We could whip out some calculators and start dissecting exponents and logs. I mean, it's all very, very tempting, but I resist. Instead, I think we should focus on how prokaryotes have sex, or how they exchange genetic information during reproduction. Prokaryotes tend to reproduce asexually in a process called binary fission. Basically, one cell splits evenly down the middle into 2. But like eukaryotic cells, more on those in our next episode, every dividing prokaryotic cell has to first copy its DNA before any of the real magic happens. It's a little easier for prokaryotes, though, since that DNA isn't trapped into a complex arrangement of multiple chromosomes. Instead, bacterial cells usually just have one singular circular chromosome, and that chromosome isn't confined by a membrane-bound nucleus. It hangs out in a fuzzy, undefined region called the nucleoid. In bacteria, all the DNA in the chromosome is held together by a set of helper molecules called nucleoid-associated proteins, or NAPs. NAPs kind of work like histones, which we've already met before in eukaryotic cells like ours. Archaea actually get to use both, either NAPs or histones, in order to keep their DNA properly condensed. But not all prokaryotic DNA lives in the chromosome. Prokaryotic cells may also have extrachromosomal DNA. These are found in small circular double-stranded segments that float around the cytoplasm like life buoys. These extrachromosomal circles are called plasmids, and a single prokaryote could have hundreds of them. They often carry genes that confer some extra advantage to the cell, like instructions for outsmarting a certain antibiotic, or for making biochemicals that might be useful in some super specific secondary metabolism reaction, or even for turning toxins from its environment into food. The ingenious design of the plasmid turned out to be incredibly inspirational to early molecular biologists. Since the 70s, we've used artificial plasmids in genetics and bioengineering to make it easier to introduce microorganisms to specific genes of interest. We call this foreign genetic information a vector, and it's almost single-handedly made the whole field of genetic engineering possible. Within a prokaryote, a plasmid can replicate itself without waiting on its host cell to divide. In biology, we sometimes call DNA like this, acting like it has a mind of its own and all. We call it a replicon. Plasmids are replicons. So are viruses and viroids. And so is the chromosome in the nucleoid of the prokaryotic cell, the chromosome that gets replicated right before the start of binary fission. But as cool as it is for things to be all independent and everything, nature is obsessed with a good old-fashioned meet cute. Sometimes bacteria hit it off and a kind of information transfer takes place. One example of this happens indirectly. It's called transduction. Basically, one bacterium accepts genomic DNA from another bacterium with the help of a wingman, a go-between of sorts. In this case, it's a lysogenic bacteriophage. Head back to our last episode to learn more about these guys. Note that when this happens, it's not as if the bacteriophage is trying to be some kind of matchmaker. Accidents happen. While a phage is getting its hands dirty infecting one bacterium, it sometimes, gets its hands dirty. A fragment of the host's DNA ends up packaged into the new virion instead of the DNA of the original virus. Then when that phage hitches a ride on a new victim, it introduces DNA from its last one instead. Another type of information transfer is a bit more grisly. We first talked about it in our episode on central dogma. It's a process called transformation. One way I like to model this behavior is to think of, Labrador retrievers. I have 2 labs, and if you spent enough time around labs 2 or any of those really big friendly dog breeds, you'll know what I mean. They're endlessly curious. They're designed to pick things up and hold on to them. And by George, they intend to do just that. Take a puppy on a walk, and I promise you're going to spend half your time trying to keep it from putting everything it sees into its mouth. They're like babies, but with tails. Yet another thing they have in common with bacteria. If a bacterium comes across DNA floating loosely in its environment, what's it going to do? Eat it. Hoard it away. It uptakes the foreign DNA and stuffs it into a chromosome, or else turns it into a plasmid. Once the bacterial cell has absorbed this new genetic information, it can start to express it, and it assumes new traits in the process. But now you ask, how does DNA manage to find its way into the environment like that? After all, isn't DNA supposed to function within a living cell? Isn't that the point? Yep, absolutely. But when that cell dies, say by initiating an emergency self-destruct sequence, it can explode. And all its guts, including, yes, its precious little DNA, ends up splattered like roadkill. So when a live bacterium comes along to scavenge for spare parts, it is kind of engaging yet another form of genetic exchange. Just that the other guy happens to be, dead. The whole thing is like some kind of self-assembling ship of Theseus, like Frankenstein and Frankenstein's monster rolled up in one. You get the idea. But there is one more type of genetic exchange that's far more collaborative and Yes, this time both prokaryotes are alive and both are working directly with each other. It's called conjugation. Conjugation relies on a unique extrachromosomal element called the fertility factor. It's also called the F factor or the F plasmid. In fact, it was the very first plasmid ever discovered. In E. coli cells, as a matter of fact, way back in the 1950s, by an American microbiologist named Esther Letterberg. Letterberg also discovered the extremely important lambda phage, one of the bacteriophages we talked about in our last episode. And she also founded the early research that would come to inform our understanding of transduction. I won't go into too much detail about how the F factor works, but for now, just try to remember this. Bacteria with the F factor are considered the donors. We say these are F positive bacteria. Bacteria without the F factor are recipients. We call these F negative bacteria. In the process of conjugation, those F negative bacteria end up receiving an F factor from the F positive bacteria and end up becoming potential donors themselves. Let's pause here for a second. Note that instead of the words donor and recipient, you may come across some resources that use the term male and female. Personally, I take issue with that kind of analogy with bacteria, since of course, prokaryotes are incapable of exhibiting any of the features we tend to associate with sex characteristics. They're microbes. It's not really that productive to make those kinds of comparisons. Instead, I prefer to exclusively say donor and recipient, or better yet, F positive and F negative. And I encourage you to do so as well. Anyway, the F factor is a great example of a plasmid acting as a replicon, since the process I just described seems to be built solely around getting more F factor copies in circulation. But the F factor genome doesn't only exist in extrachromosomal form. In some cases, the F factor is directly integrated into the bacterium's chromosome. When this happens, the prokaryotic cell is designated the term HFR. That stands for high frequency of recombination. When this HFR cell performs conjugation, the F factor gets passed off to the recipient like usual, but so do standard bacterial genes, and thus old and new bacterial instructions become recombined. The recipient cell now has a genome consisting of recombinant DNA. Oh, and I almost forgot the most important part. How does the F factor make it from one cell to the other? Through special pili called sex pili, which I mentioned a little earlier. A sex pilus from the F positive cell extends toward the F negative cell, and a special structure called a mating bridge forms between them. Once contact is established, the pili contract, and both cells are pulled inward toward each other to facilitate the movement of genetic information. The two, briefly, become one. Behavior like this helps to explain why the whole male and female analogy tends to be so attractive to many biologists and why it might be an attractive analogy to you too. But again, resist that. With the exception of binary fission, all the genetic exchanges we've discussed up until now are not procreative. No new bacteria are formed. And even with binary fission, only one prokaryotic cell actually gets involved. Again, prokaryotes are asexual organisms, period. Any other movement of genes represents a type of horizontal gene transfer, that is, from one member of a generation to another, not vertical gene transfer from parent to child. Horizontal gene transfer does nothing to increase the bacterial population. Its only evolutionary usefulness is that it has the potential to make the existing population more diverse, and so, in theory, more resilient in the face of enemies without and within. We've kind of gone all over the place in this episode. We've talked about prokaryotic structure and morphologies. We've talked about bacterial classification using the Gram-Stain technique. We've talked about adhesion and motility. We've talked about infection and virulence and endospores. We've talked about prokaryotic reproduction. We even talked about prokaryotic kind of, sort of, not really reproduction. But in all this, we've managed to avoid the biggest elephant in the room. There are two domains of prokaryotes. Bacteria is one prokaryotic domain, and boy, have we talked about bacteria. But the other domain comprises a set of organisms we collectively call the Archaea. I've mentioned the name several times in this episode alone and a few times before. But what exactly are they? Why do they only get passing remarks? After all, there are only 3 domains of life to pick from. 3 domains of life on Earth, 3 domains of life in the whole of the observable universe. Eukarya, bacteria, Archaea. Why does one always get the short end of the stick? One answer might be that it's the domain that's been most recently discovered. We've known about eukaryotes since the human race has ever known about anything. I suppose we're eukaryotes, so for most of human history, almost every living thing we've ever been able to see with the naked eye has been a eukaryote. And as for bacteria, we've known about them since at least 1676, thanks to an inquisitive little Dutchman named Antony van Leeuwenhoek and his brand new invention called the microscope. But Archaea weren't discovered until 1977. In autumn of that year, 2 American scientists, a biophysicist named Carl Woese and the biochemist George E. Fox, published a paper setting out their argument for why a certain group of methane-producing prokaryotes should be reclassified. Reading it, you're struck by the same cogency and directness that you might have felt after reading Watson and Crick's paper on the structure of DNA, or even Brenner, Jacob, and Messelson's paper describing the structure of mRNA. It's short, barely 2.5 pages. It's clear and succinct. It gets to the point. And it's a point that anyone can understand, no matter whether you do science for a living or not. I'm going to read you just a little bit of the opening so you can see what I mean. It goes, "The biologist has customarily structured his world in terms of certain basic dichotomies. Classically, what was not plant was animal. The discovery that bacteria, which had initially been considered plants, resembled both plants and animals less than plants and animals resembled one another, led to a reformulation of the issue in terms of a yet more basic dichotomy, that of eukaryote versus prokaryote. The striking differences between eukaryotic and prokaryotic cells have now been documented in endless molecular detail. As a result, it is generally taken for granted that all extant life must be of these 2 basic types. Thus, it seems that the biologist has solved the problem of the primary phylogenetic groupings. However, this is not the case." Woese and Fox go on to suggest that the class of methanogenic microbes then considered bacteria metabolize in a way that just doesn't make any sense under that classification. They note that these bacteria do have cell walls, but unlike most bacteria, they don't have peptidoglycan. They note that these organisms process energy, but to do so, they rely on completely unique coenzymes. And finally, in comparing sequences of ribosomal RNA present in eukaryotic samples, bacterial samples, and methanogenic bacterial samples, they note that this final class is as much similar to bacteria as the bacterial class was to baker's yeast or duckweed, or the cells of the gut or the pancreas. In other words, not that similar at all. No, these were something new, or in a way, old. They called them Archaea bacteria, which from Greek literally meant primitive bacteria. Today, we've discovered many more of them and often just go by Archaea. The whole primitive label stuck because The unique metabolism of the first archaea that were discovered suggested that the whole domain was built for a planet that no longer really existed. Not completely, anyway. A rough, inhospitable landscape, a playground for extremophiles. And yes, many of the Archaeans we've discovered since then definitely thrive best in those pockets of Earth that still feel near apocalyptic. Places like Yellowstone, with its hot springs and thermal vents, are perfect breeding grounds, both for extremophile bacteria like the kind Tom Brock discovered, and for extremophile archaea, like the ones that were later named after him. Nevertheless, archaea are abundant in less extreme environments too, in the soil, in ocean waters, even in your gastrointestinal tract, alongside all the other prokaryotes that make up your microbiome. So to return to our first question, why don't we talk about them more? If they're everywhere, and if they've been everywhere for ages, why might this be the last time we'll ever give them a spotlight? Unfortunately, they're a real pain to study. In order to make meaningful observations and run controlled experiments on microscopic life forms, we have to be able to culture them in rigorously managed, easily reproducible conditions. That's the essential rule that makes the work that I and other cell biologists do possible. But thanks to their overwhelming predisposition for extreme environments, it's next to impossible to accomplish that successfully for Archaeans. That was the genius of Dr. Brock. He did a lot of his work in the field, right in the thick of it. He did science the good, messy way. And while that approach will only take you so far, it'll definitely take us farther than we are now. There's so much we still don't know about Archaea. Can you imagine what we could do with those insights? If nothing else, this much is striking. 1/3 of the entire story of life in the universe is missing, virtually unknown. Maybe it is time for us to shop on some boots, to take some detours, to go where the life is, and get curious again. Thanks for joining me on this whirlwind tour of all things prokaryote, and a special thank you to the incredible folks at the University of Wisconsin-Madison who kindly shared that audio excerpt of their 2017 interview with Dr. Thomas Brock. While Dr. Brock passed away in 2021, his work and legacy continue to resonate in the ongoing research of scientists just like him, all committed to the quiet, tedious, lifelong mission of discovery on scales both large and very, 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 very small. Now is a particularly critical moment for academics, for researchers, and for the numerous caretakers of natural spaces in the United States and around the world. Take a moment today. In fact, let's do it right now together. Just a moment to reflect on exactly what sort of world these folks have made possible for you, for me, for the people we care about and the things we value. And now, just stepping out of that moment into a world you get to help make too. As always, I'm Michael. This is Curiosity. See you soon.